Good morning, everybody. My name is Yamhilette, and I'm going to present some of our work using the Isoplexis technology available in the M Center. The title of my talk is Functional Phenotyping of Glioblastoma-Associated Macrophages and Microglia using Isoplexis technology. First of all, I just want to acknowledge my team. I'm a part of an amazing laboratory guided for Dr. Piccirillo. We have the luck to be collaborating with a lot of departments in UNM and outside UNM. And also we have the privilege to work with patient-derived samples. So we are extremely grateful with the patients and their families. And also I want to acknowledge all the funding and support that we have. Okay, so first I'm going to start talking about glioblastoma. Glioblastoma is an extremely heterogeneous and extremely aggressive disease. It's the most aggressive primary brain tumor, and the standard of care that is available right now it's based on something that it was really important, but it has like almost 20 years. And even if it's like the best that we have now, the 7% of patients survive 5 years post-diagnosis. And one of the reasons for this is that it's highly heterogeneous, and that's why it's so important to find out ways to work with this heterogeneity of this disease. One of the ways that has been designed to be able to get samples of glioblastoma is using a fluorescent guide multiple sample scheme. And this is based on the use of a drug called 5LA. 5LA is given to the patient before surgery. And what it does is it goes into the tumor cells, and under blue light it fluoresces. This helps the surgeon to do a better resection, because as you can see here, what I'm trying to play now is a video courtesy from a neurosurgeon that is a collaborator, and you can see a part of how this looks in real life. Thank you. As you can see here, this is a surgery. You can see the blue light, and you can see how under blue light, when a patient is P5LA, these fluorescence help the surgeon to do a better resection. Okay, Dr. Piccirillo has worked with a lot of groups trying to come up with a better model that can help to have this heterogeneity of the samples in the laboratory. As you can see here, and again, I'm so sorry. As you can see here, having 5LA, as I mentioned, it helps to have like better tumor resection, but also the design of multiple sampling, meaning inside of the same tumor take several samples instead of one, that has helped to increase the spatial resolution of samples, which better exemplifies the heterogeneity of the disease. So this is why this is like the base of a model, and this is gonna come up with what I'm talking about, why it's so important. From the samples that we take from surgery, and we dissociate the tissue, and then we culture those cells. Those cells are not they are not under any enzymatic digestion. They don't have any cell sorting, and it has no or limited in vitro passaging. So what we're trying to do is to come up with a model that better comprises what is happening inside the patient. Now, the laboratory has a sizable GBM patient sample collection, and what I'm showing here is how many samples we have so far. This is the number of patients, and so far we have 30 patients, 153 samples. And as I mentioned, because it's a multiple sampling, we have several samples of each. So we have 96 tumor masses, 23 SBC, and 34 margin samples. Now, these are not only allow us to investigate the tumor cells in the samples, but also it allows us to understand the tumor microenvironment. The tumor microenvironment in this context is any cells that is not tumor cells. And we are interested in understanding the tumor microenvironment because the context of the tumor is very important of how it grows. Now we know that how the tumor microenvironment and the characteristics that it has can prevent and can have like anti-tumoral effects or can have pro-tumoral effects. Meaning, and this is true for the architecture of the tissue, where it's outside that are not cells, but also the phenotype of the cells that are outside that are not specifically tumor cells. What we interested in this project is in something that is called TAMs, or tumor-associated macrophages in microglia. Now, in the case of the brain, the tumor-associated macrophages are the ones that are from monocytes that are from the bone marrow that travel to the brain, but also there are microglia present that is brain residents. And these two have capabilities that can be tumor supportive, which means that they're going to have like inflammatory effects that is going to allow the tumor to grow, or they can have anti-tumor effects and they can prevent the tumor to grow. So it's very important for everybody to understand what is the nature of this, or if there is some forces that makes them change. Now, that's why this is the base of our study. We, as I mentioned, we have this amazing technique that allows us to take samples from patients. And what I'm doing is isolating these TAMs, these macrophages and microglia presence, and throw them in culture. Then we want to run a single-cell transcriptomics because that is going to give us the map of how they nature. But something that I come up over and over again is the importance of functional phenotyping. It's not enough to see like the transcriptomics. We have to know how they are acting at a functional level. And we are also interested in knowing if after treatment that mimics the standard of care is going to change, is the nature of this is going to change, are they going to function different? And this is how this technology Isoplexis comes in, because this has the capability to help us quantify cytokines in real time in our cells. Sharina mentioned a little bit about Isoplexis. I'm going to not go very into depth of how it works because I know we have another talk about it, but as summarized, this is in here I'm showing the single cell isocode chips. Here you put your cells, and they have a functional proteomic barcode that is going to detect the release of cytokines. The way that these chips function is that you buy the chips that have the cytokines that you are interested in, or the pathway that you are interested in. And there are usually like several, and we work with the human single cell innate immune for now. Now, this machine was like, it arrived in the end of December, and I was like trying like a week before the break. But before, we were working closely with the company and we were sending our samples to them and they were running these analysis. So what I'm going to show you is kind of like a mixture of both things. We require to do like a lot of troubleshooting, both their team and our team. And one of the reasons is because our cells are very different and they are very heterogeneous. And as to our knowledge, this is the first time this technology is being used for something that is similar to our model. So most of what I'm going to show you is speaking about the troubleshooting and also advice that I have of people that want to use the technology and why I'm so excited to use it. Okay, first I'm going to talk about the codeplex secretome, which is the other use of the technology. The codeplex is kind of a very high sensitive ELISA that you can run a lot of cytokines at the same time, like 30 cytokines at the same time. But also it requires a tiny bit of sample, and that helps us a lot because our samples are like so precious and they were very different from other models. You only need like around 12 microliters per sample. It's 2 repetitions and they need like 5.5 each. So if you have like a teeny tiny sample, this is going to detect it. In this case, in the codeplex, one chip allows you to do 8 samples. So you can run one experiment with one chip. And then you can read the results. In this case, I'm showing you again the human innate immune cytokines that we were interested in analyzing. And when we were working on this, and yes, this is another diagram of the chip. These are the 2 repetitions, the blanks, and it takes a little bit of training to understand how to load the chip. I think that's like one of the hardest parts of how to learn how to do this, but it's not that difficult. One consideration that I should mention is that some media that is used for tumor-associated macrophages and microglia have cytokines. And we were testing our media using codeplex, and we also were testing using some external cytokines because we wanted to know what was the sensitivity. And we saw that when in the program, I should mention this, when it's like not real color, like this compared to this means that it's below the threshold. But we noticed that the media had some detectable levels of cytokines. So we also test that, and is my recommendation that to do that when you're working with this technology, because some of the medias are expected to have cytokines because they are specific for growth of these cells. In particular, this promocell M2 media has a cytokine cocktail, but they don't tell you what is in there. So you have to make sure that the cytokines that you're testing are, if they are from the media, you have to take that into account. In this, what I'm showing you here is some of the results that the program gives you. You can have the concentration in the picograms per milliliter. And these 2 media from bromocells show detectable levels of some cytokines. And another media that is from another company, Licon. And also I use the RPMIS as a control because it's used in another experiment and I get found. So one of my advice is to also test this just to make sure that if you're using a specific media that is not like a common media, like a biomedical, that is a media that is specific for, in my case, tumor-associated macrophages in microglia, just test and make sure that the cytokines that you are interested in are not going to be affected by the ones that are in the media. As Dr. Piccirillo was saying, our main focus is using this part, which is the single cell secretome. Using again this diagram. In this case, we use cells, not media, and those cells are going to go into chambers that are going to detect the secretion of different cytokines. And again, you pick the cytokines depending on what you want to study. There are like several different shapes that have like a different panel of cytokines. This is the one that we are interested. Again, the human single cell innate immune. They also have panels for mice. One other consideration is that in these experiments is one sheet per sample. So you use one, in the other one was 8 samples for one sheet. In this one, you use one sheet every sample. And the protocol asks for a little bit under million cells, but what you load is this particular point. Okay, so this required a lot of optimization, as I mentioned. There is not a specific protocol for glioblastoma-derived terms from human. So the closest is this human monocyte protocol that the company gives you. One thing that I like about the protocols that they give you is they have a lot of details. The protocol is very detailed. It tells you where they got all the instruments, all the media, everything that you need. They also have like catalog numbers for everything that they use. And this is a diagram of this particular protocol that I had to optimize for our purpose. First, we have, in this case, you can like toss cells, which was done when we were shipping our cells to the company. So they have to thaw the cells and then grow them and recover. In my case, I have them in my incubator and I enrich them in 4C11 positive, which is going to have the macrophages and microglia. Then in this step, we don't use it. We don't really stimulate the media. We just compare different. We don't use a stimulant to make them produce cytokines. We want to know like how are they measured. So we are using different media to see how they behave, but really we don't use this step unless we're using a control that is stimulated by PS. This was a step that was particularly difficult for us because our cells is not a cell line. So to get to numbers that are like this big, it takes a lot of time. And as we mentioned at the beginning, we want to limit the passage number. So we kind of push it a little bit and work with less cells and see how it looks. And also we have less cell available than others, and the growth rate varies between patient to patient and also between the samples of the patient. So to get to a point and have like around the same amount of cells that grow slower, it's kind of difficult. So this was going battle, and also the company going to sell the cells in part because these cells are very hard to grow, but also because they take a long time to recover after thawing them. It was hard to use them, and sometimes you start with a size of a run of cells, but at the end of the process, they don't like it. So I was able to use less cells and get decent results. So in one part of the protocol, in one of my experiments, my cell count was 0, and I still see some cells because I didn't grab, like it had like almost nothing. But if there are cells there, and they are detected by a machine because it's very sensitive. This is how, like more or less how it shows in the machine. you write down the names of your samples, and it gives you the number of quality cells and also an IsoQ score. And I'm gonna talk about more about what it means. But if it says high, it means that the results are like representative of what you put there. If it's low, you have to go back and see like, okay, how the sample looked, were the cells dying, or you didn't have any cells. Okay, so this is a little bit how the program that the machine use looks. I'm using this Isospec 290, and it allows you to see so many different ways of the same data. It gives you like, you can see protein frequency, signal intensity, distribution, and polyfunctionality, which means that some cells have the capability of these different cytokines. If you have a lot of cells that are polyfunctional, it's going to tell you. And this, I'm just showing you one graph as an example. This is measured in relative fluorescent units. And here I'm using like control cells with, this is my panel. And each one of these dots is gonna be a cell, and this is the intensity of each one of the cytokines. I tested the macrophage control, which is like these blue dots, which are this isodonor and this activated isodonor, and as expected, isodonor, which is a cell line that provides the company, and a lot of the protocols are optimized for this. So it's a good internal control if you want to make sure that something in the process, like if this doesn't work, it means that something in the process didn't work. But if this works and you don't see anything in your samples, that more likely means that it's your samples that don't have cytokines. And as expected, these activated isodonor cells that have higher higher intensity of these cytokines. And when I run this experiment with my sample, it looks something like this. And for this other example, I'm using secretion frequency, which is what percentage of the samples had the released one different cytokine. And again, I'm using this macrophage cell line versus these 3 sides of the same patient. And you can read this like in so many different ways, and you can present this in so many different ways. Here, I can try to understand the difference between one region of the other versus the same patient. I can have information about what cytokines have all of them in common. You can have like a lot of insights, and from this information, users for other experiments can have more insight of what your macrophages or microglia is doing in your samples. And it can get a very bit overwhelming when you get like all the data at the same time. It can be presented in different ways. I can compare one to the other. I can compare these 4. These are 4 from an 8 sample experiment, so it was by force. But the sensitivity is very good, and we are really excited to keep using this now that we think it's more optimized than it was months ago. And I want to finish my talk with this. And I don't know how I got this picture, but this is like a picture that in one of the first experiments, how these microchip look, and these were the cells moving through the chip. And my take home is this one. I have to give you one idea. Is this one. Including functional assays in translation and research is hard. It's going to be hard. It's expected to be hard, but it's also very important. It's important to do this work. It's important to optimize this work, because this is how we get the information that is important for the patients. And that's all I have. Thank you so much.